Yes, of course. Right as I start recording. Hello, you cat. Sorry, the little ball is still spinning. Cat, why now? Okay, right. Okay. Just as I start recording, I get entangled with a cat. Nice cat. There, you can stay there. I've given you another set of practice problems, and I'm going to work through one of them. Just again to try to help you work out how to do these things. It is a skill that benefits with practice. So let's start by looking at the practice problem sheet. This first one is actually very similar to one on the first problem sheet. There's a number of cases where you'll get pretty much the same problem, but it's done with different organisms. But the second one about mice is one that you haven't seen yet. A gene found in mice determines whether or not any pigment will be produced in the fur. The big C allele is dominant to the little c allele. Mice with no pigment are white. A second gene determines how dark pigment is distributed. Some mice have agouti fur and others have solid colored fur. Now, unfortunately, our tabby cat is not available for consultation at the moment, but you'll actually see this if you have a striped cat, a tabby cat. If you look very carefully at the hairs that make up the lighter colored stripes of a tabby cat are mostly light colored, but they'll have dark tips and usually dark bases. That type of pattern is called agouti, and some mice have agouti all over the body. A few cats do as well. Typical tabbies have bands of agouti fur and non-agouti fur. But a breed of cat called Abyssinian has agouti hair all over. And some mice have agouti fur all over, and other mice have solid colored fur where all of the hair shaft has color. This particular one has white spots, but his dark hairs are dark all the way through. Whereas if you have agouti fur, it's got kind of a mottled heather gray appearance to it. I'm not perfectly sure how to describe it, but it's easy enough to see if you've got a cat handy. Okay, speaking of cat handy, you can get off right now. There, good kitty. Right. So we have two genes that we have to take into account, one of which determines whether we have pigment at all, the other of which determines whether the pigment is all through each hair or only at the tips and the bases of each hair. So when two different genes interact to determine a phenotype, we call this sort of thing epistasis. That's just a word you need to know. And we have the question, what are all the possible genotypes of an agouti mouse? So here's the board. What are the possible genotypes of an agouti mouse? Think about it. If you have a mouse that doesn't have... Okay. If you have a mouse that doesn't make any color at all, it can't be agouti. It can't be solid. It's only going to be white. So for a mouse to be agouti, it's got to have one copy, at least, of that dominant allele for color. All right. It can't be little c, because then it's just going to be white no matter what. But it could have big C, or big C, little c. Both of those will be a mouse that has color in the fur. So we can write big C blank, meaning one allele is the dominant colored allele, and the other one, it doesn't matter what it is. We also know that agouti is dominant to solid color. So the mouse has to have at least one allele that is the dominant agouti allele, but it doesn't matter what the other one was. So if we want to write out all the genotypes of an agouti mouse, it could be homozygous dominant for color and homozygous dominant for agouti, or heterozygous for color and homozygous dominant for agouti, or homozygous dominant for color and heterozygous for agouti, or heterozygous for both. Those are the four possible genotypes that will give you the phenotype of agouti fur. Now, let's see. The question is asked, if two white mice mate, can any of the offspring have agouti fur? You can do a Punnett square if you are not sure about this, but a white mouse is going to have genotype little c, and then it doesn't matter what the agouti alleles are. It could be little c, big A, little c, little a, little c, big A, little a, it doesn't matter. So if you cross two mice that are both white, it doesn't matter what agouti alleles they have. All of those offspring are going to get only a colorless allele from mom and a colorless allele from dad. That's all that the parents have to give them. So all of the offspring are going to be colorless. It'll be white. And it doesn't matter whether they've got agouti alleles or not. You will just never see them. Okay, so hopefully that's fairly straightforward, but let's go to the problem. Suppose we cross two mice with the genotype big C, big A, little a. What's the result of the cross? Well, we're crossing one parent that is big C, little c, and I'm going to do that little c with a little cursive loop, just so you can tell them apart. Here's big C, big A, little a. And the other parent has the genotype big C, big A, little a. So we'll draw our square. That's a little bit too close. Just a second. All right, we'll draw our square. All right, this one should be fairly straightforward to set up. The trick is going to be interpreting it. Remember that each parent passes on one of each pair of alleles, but not both. So it can pass on the first of each pair, or the outers of each pair, or the inners of each pair, or the last of each pair. So there's good old FOIL. So this one can pass on big C and big A. It can pass on an allele for color and an allele for agouti. Or it could pass on the allele for color and the allele for not agouti, solid. Or it could pass on the allele for no color and the allele for agouti. Or it could pass on the allele for no color and the allele for spotted. And with this one, it's the same thing. So we've got a 4x4 Punnett square. So of course we draw the lines. And there we are. Okay, so we just got to fill these in down and across. And so here we put big C, big C, big A, because we sort the letters. That corresponds to a mouse that has color in its fur. So that mouse will be colorful. And it's homozygous dominant for the agouti phenotype. So that mouse is going to have agouti fur. It's going to have this kind of mottled heather gray color to its fur because it's got color and it's got agouti. And this one will also be agouti. And this one will also be agouti. And this one will also be agouti. Sorry about my rotten handwriting here. I hope you can make it out. This one will be agouti. That's an A. This one will have color, but it is homozygous recessive at the agouti gene. So this one is going to be solid. That one's going to look black. That one will not have the agouti phenotype. This one is going to look agouti because it's got one of the color alleles and one of the agouti alleles. But this one is also going to look black because it's homozygous recessive at the agouti gene. It's got 2 copies of that non-agouti allele. So far we've got 6 agouti and 2 that are black. That one's going to look agouti. This one will look agouti. Come on, Wagner. This one here is going to be white because it's got 2 copies of that recessive allele for no color. And it doesn't matter in this case whether it's got agouti alleles or not. If it doesn't make color, it can't show an agouti or a solid color phenotype. You know, if it doesn't have the ability to make color, it doesn't matter what other genes it's got, it's going to look white. So that one's going to be white. This one is going to be white. And this one will be agouti. And this one will be not agouti. This one's going to be a solid color. And this one is going to be white. And this last one is going to be white. So if you do the count, we have 1, 2, 3, 4, 5, 6, 2, 3, 4, 5, 6, 7, 8, 9 that are agouti. So 9 out of 16 of the mice that you cross will have agouti fur. That's a little bit more than half, what, 56%. And 3 of the mice are going to have solid fur, 3 out of 16, which is what, 18%, 0.75, 19%, I think. So that one, and that one are going to have solid color fur. And then these 4 down here in the lower corner, those 4 are going to have white colored fur. So you're going to end up with 56.25% of your mice are going to have agouti fur, and 3 out of 16, or 18.75% of your mice, are going to have solid color fur, probably black, and a quarter of your mice, 25%, are going to have white fur. So in this case, you get a ratio of 9 to 3 to 4. In the problem that we worked in lecture, what you got was a 9 to 3 to 3 to 1 ratio. And I've had students before who kind of assume that every time they see a dihybrid cross, the answer is always going to be 9 to 3 to 3 to 1. And I've seen people put down this answer even when it's very far from the truth. But you can't memorize what you think the answer is going to be. That would be like when my mom was in high school struggling with algebra. She was known to say, why couldn't they just all get together and decide that X was always going to be 7? Wouldn't that make things a lot simpler? She was joking. But yeah, X is not always equal to 7 in high school algebra. And genetics crosses don't always get you 9 to 3 to 3 to 1. Sometimes they get you different things. There are other patterns you can get. So let's see here. The third one, the first one is actually a type you've seen before on the practice problems. And the third one, you might actually want to wait until I've gotten to talk about enzymes. I may end up posting a third round of these just to make sure everybody's got plenty of practice here. So stay tuned for that. But that second one is one that I wanted to work to show everybody how epistasis plays out when you're doing Mendelian genetics. And that's not the only possible pattern you can get when you have epistasis going on. Okay, I'm going to go ahead and stop recording right now. I hope this was somewhat helpful. And we're done.